就the microphone is on the computer. Can you change that? How are you guys feeling? Okay, alternatively, restriction. Okay. Clean it up. Alright. So we have recombinant DNA. We already know that recombinant DNA is essentially the modified DNA that goes and gets inputted into another organism, right? So yeah, bacteria are the most widely used genetically engineered organisms, and this process is used to produce from bacteria are common to all genetic modifications. So, as in when we genetically engineer bacteria, we're going to be changing their genome by putting in new stuff or removing some stuff, etc. Now, the basic processes used to produce these from bacteria are going to be common for all modifications. So when you use them in bacteria, you're going to be able to use them in other organisms as well, like rats, rabbits, cats, bats, gnats, while they're in flats, sitting on gnats. I'm make sure you gotta make sure that you got like bats and no gnats and like a So, different copies of a desired gene can be made by taking an mRNA molecule transcribed from the gene. Shut up! We're taking an mRNA molecule transcribed from the gene. So we have, okay, so let's say we want a gene, and we want this gene. We want this desired gene. Why do we want it? Because we want to put it into another organism. Let's say. So let's say there's a gene for producing an enzyme or protein A, which isn't present in human, or which isn't present in the bacteria. So if we want to put it in there, then we need to get it. How do we get it? We need to get the gene. We need to get the gene so that the enzyme can describe it or translate it, right? So now we can make an artificial copy of this desired gene. Now remember, if I wanted to make an artificial copy of the gene, that means we're trying to make DNA. Now, why not just take the DNA and put it into the organism that we want to put into a bacteria? The problem with that is if you just directly put it in, that's going to have introns in it, exons and introns in it, which we're going to have to remove. Now, bacteria don't have DNA, no, it's RNA. It doesn't have RNA splicing, right? So, we need to use the already post splicing or mRNA molecule. Okay? And we're going to be using that to produce the correct DNA sequence, kind of like reverse engineering. And when you do that by using a reverse transcriptase enzyme. So that just reverses the transcription process to get that complementary DNA, which is just basically your the strand of DNA before being turned into RNA, the original strand, I guess. Alternatively, restriction endonucleases are going to be used to cut DNA strands into small pieces that are easier to handle. So let's say the gene is just like really, really long, right? Let's say the protein, I mean, it could happen that the protein is going to be super long. And so to combat this, we're going to try to make the DNA strand of the gene shorter by using restriction endonucleases. Now, each type of endonuclease is going to be will cut the DNA only at specific restricted sites within a particular DNA sequence. So, like how there's going to be specific sites for stopping and starting, there's going to be specific sites for binding of the transcription factors. There's also going to be specific sites for the restriction endonucleases to cut the DNA into pieces, right? So some restriction endonucleases can only cut in a way where it leaves a few base pairs longer on one strand than the other, so it's not equal. This is called having a sticky end. A sticky end makes it easier to attach new pieces of DNA to them. Oh. So sticky ends attach to other compatible sticky ends. So, for example, you can join together sticky ends of DNA fragments cut by E41, but not sticky ends cut by two different enzymes. So if we have an endonuclease called E41, right? So those are only going to be creating sticky-ended DNA fragments. And so those DNA fragments have an easier time attaching together compared to another, fragments cut by other endonucleases. And then the next step is to integrate the new gene into the vector. Plasmids, the circular strands of DNA found in a bacteria. Okay, man. Next step is to integrate the new gene into a vector. Plasmids, the circular strands of DNA found in bacteria, is frequently used as a vector to carry the DNA into a host bacterial cell. So, let's say we have the isolated or the cloned DNA gene, DNA of the gene. I mean, a gene is DNA. Anyways, we have the gene which is isolated. Now we want to somehow get into the bacteria, but how would we do that? We use that using the plasmids that are found in bacteria. So we're going to be using the plasmids, and we're going to be using that as a vector to get it into the DNA of a bacterial cell. It's funny that because plasmids are also found in other bacteria, right? Anyways, so DNA ligases are used as the genetic glue to join pieces of DNA together. We already know this. There's also DNA integrase, but we're going to see that later. We're going to see in figure A, DNA ligase is going to be used to put it into the plasmid. Now we cut the plasmid with the restriction in the nucleus? Oh. I see. So we talked about how restriction endonucleases are used to cut DNA. I thought that was to make something easier. Actually, okay, well that makes sense. It's gonna be used here, right? I mean, I hope that makes sense. I mean, like, we knew what it could do, but I didn't really know why we were using it, why we would use it for the original gene. But hey, I mean, So we're using it in both cases actually. We're using it here because it has a sticky end. It has a sticky end over here. And so if we had another, let's say we broke it down here, so that sticky end would bind to this pretty easily. Or maybe if it's the same restriction and the nucleus, then when you cut it, these are also sticky, so these bind on pretty nicely, right? So it's like you can have a lot of possibilities. Oh, baby. So, okay. Damn, bro. This shit is so good. Huh. So sorry.
Should not leave this earth unfinished business. He should live each day as if it was a pre-flight check. Should ask each morning.
Question. I'm sorry. I'm having too much fun. Okay. Gorilla Glue. Okay, so what are we doing again? Something about biology? I gotta study, man. Okay, so we're using plasmids. Plasmids are found originally from DNA, but we're going to be using them to carry that isolated gene into the DNA of a host bacterial cell. The DNA ligase are going to be using as the genetic glue to join pieces of DNA together. This is how we're going to be inserting that chosen DNA cut from an organism or made artificially into another piece of DNA that will be carrying the whole cell. Once the plasmid is incorporated into the host nucleus, um, sorry. So the plasmid is what we're using to convert it. So we remove the plasmid from this cell and we put it in. This is the original bacterial cell. So we put the plasmid into the bacteria. So we don't put this into the plasmid of the host bacterial cell. We put the plasmid inside the host bacterial cell. Now it's okay, you can have it because plasmids they're found naturally inside bacteria, but not in all bacteria. Or maybe some bacteria can hold more than one. Either way, once that plasmid is incorporated into the host cell, it's going to be forming a part of the new recombinant DNA of the genetically engineered or transferred organism. Now, officially, you can say that the bacterial host now has genetically modified DNA, right? And so um, now we can consider genetically engineered or transformed organism. Okay? Now, in this bacterial host, it remains an autonomous plasmid capable of independent replication, as bacteria do not have a true DNA. Successfully transformed cells can be identified, isolated, and cultivated on an industrial scale, so the proteins that they make can be harvested for human use. A successfully transformed cell can be identified, isolated, and cultivated on an industrial scale, so that the proteins they can make, or they make because of this thing that we made, is harvested for human use. This technology is developed constantly, and gene editing is becoming easier, cheaper, faster, and more sophisticated. Can I gene edit at home? I'm obsessed with biochemistry and gene editing. I read about it every day, and I really want to try it myself, even if it's not plant or just leaves. The odin that sells as a PhD scientist within being for 12 years, going on 15 years, I get the appeal of trying to do this at home. Like I kick out of doing it for my job. The idea of doing this at home seems really hard because it's really hard to get things to work well with ideal settings and tools. And certain people like these really give me a bad feeling, not because I think it's randomly helps. Actually, if you're this much into biochemistry and medicine, you're able to try to write some nearby academic labs to see if you can get someone to bring you on and mentor. Maybe you don't live in it, and even if you do, it can still be a long shot, but it's worth thinking of people with this word. When I told him I was in high school, he said artwork, is like, I just dropped by spring break. Not doing this, that's not what they're for. You can really do engineering on cells and culture, but doing that requires tissue culture setup, DNA prep, experiment. Just being interest, I'm sure you'll see. Did he ever do it? The Lewis? He's gonna be 25 now. across the level at the psychologist to get understand the main ideas of the field first as professor to pick up the basic ideas of the field to the level of a graduate student engage such weeks this way they wanted to and they have found some. Well, is it gene editing? I mean, I guess it is. Look at this profile. Such a funny picture again. God, man, shut up. One game. The rhythm there. You told me to get a small cake, and the small cakes were all there, 950, 900, 900. Oh, yeah. That's how it be. Money man, money talk, money walk. Okay, so oh. Man, biochemistry, huh? Imagine I study biochemistry and become an evil doctor and I kill everybody. I'll do it. Oops. That's how organisms are made of what then responds to this? The host system is the autonomous plasma capable. So this gets read or transcripted and translated, I believe, by itself. Okay. So it has independent replication. Oh, no, sorry. It gets replicated independently, not translated and transcripted, but just replicated. This has because that doesn't have a true nucleus, which gets like the main site, which gets replicated by the cell cycle. So it has like maybe one nucleus with its own cell cycle, maybe another nucleus with its own cell cycle, but these aren't nucleus, these are plasma. The point is we're gonna be fine, like if we do this properly, then we're gonna be having a bacteria with enzymes. With plasmids, with the kind of plasmids with the kind of thing that we want, with the kind of genes that we want, so it can produce proteins and things that we want. So we can harvest them, or we can grab a bunch of these guys, right? We can just grab a bunch of these guys. Now we have a bunch of these guys making all the proteins that we want. And we collect all of these proteins, and we can sell them and make money. Money, money, money. Using bacteria to make money. I love using bacteria to make money because we make making money. I like making money every day. Money, money, every day. Using bacteria to make protein that I can harvest and use for money. Hey, 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 give me that money. Hey, hey, give me that money. Give me that, 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 give me that. Money. Easier, cheaper, faster. Easier, cheaper, faster, faster, faster. Central dog, central dog. Oh, give me that money. Give me that money.
Oh, give me that money, man. Give me that money.
Okay, so what's going on? So stage one, we have the gene in some kind of organism. We want to get the pure DNA form. So what do? We get the mRNA of the gene. After getting the mRNA of the gene, we do reverse transcriptase and we get the actual gene. Right? So this is called the cDNA or something. And so this cDNA, we're going to be using restriction enzymes to get out the yummy juicy gene. And the yummy juicy gene is going to be put into the plasma. So where do we get the yummy juicy gene? The yummy juicy plasma. We get the yummy juicy plasma from another DNA, which we're going to be extracting somehow. And our plasmid DNA we get from another bacteria. We're going to be cutting holes open in the plasma the same way we extracted the gene, the yummy juicy gene. And we're going to be using the DNA ligase to kind of, just glue it and, So after we use DNA ligase, bam, we got a fully whole, thingy. And then we just input the plasmid into the host cell, and the modified bacterium will now produce a different protein as the new gene is expressed and causes the synthesis of the protein. I will just ask. Dried and manipulated on its own. When you say on its own, does that mean it follows a separate, perhaps faster cell cycle compared to the cell cycle of the host cell? Yeah. What they tell? What they tell you, man? Hmm. 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 Okay, go. Hmm. Okay. 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 So let's quickly see what it's telling us. My cat. What are you doing? Yeah. So it's what it does. Lot of red. Oh, lot of red. Rating that. So the distinction between the host cell's chromosomal replication and the plasmid activity is that a fundamental concept in molecular genetics that clarified the plasmid is physically independent but mechanically dependent. Does it mean to transcribe and translate on its own? The plasmid contains the genetic blueprint but does not possess its own machinery. To utilize the host bacteria and the existing research to fix the gene, the host's RNA polymerase binds to the polymerase and produce amino, and the host cell's tRNA and the amino acids are also going to be used to synthesize the protein for the translation. While the plasmid provides instructions, it is entirely reliant on the host cell's metabolic infrastructure, aka the machinery of the host cell, to produce the harvested protein. That's why we're using the host cell. Autonomous replication versus self-replication. When we describe a plasmid as autonomous, we refer to its origin of replication. This is a specific DNA sequence that will allow the plasmid to initiate its own replication. So if they have a specific DNA sequence that allows its own duplication, plasmids do not necessarily wait for the host cell to begin its own binary fission. They can replicate in multiple times within a single cell cycle. This results in what is known as a high copy number. A single bacterium might contain hundreds of identical plasmids because there are so many instruction manuals of plasmids available The ribosome to read simultaneously the cell to produce the desired protein at a much higher velocity than if the gene were integrated into the whole new chromosome. So essentially what we're saying is, since that plasmid gene is replicating it autonomously, now we have loads of plasmid and we have one ribosome. And since, like, let's say there's 100 like, we can just say that the concentration of the plasmid is higher, and so the chance of the protein, the desired protein, to be translated is going to be higher as well. And so since it's higher, therefore we can harvest it more commonly. It's not question of fact, but this is what I mentioned. Once the plasmid is incorporated to the whole cell, it is important to note for exams that bacteria prokaryotes do not have a nucleus. Plasmid simply resides in the cytoplasm. The text correctly clarifies this a sentence later, but to show you don't use the form of describing the field and beautiful. I'll go with the look at this. We already went to this. Now, we said that we have to identify them, or we have to kind of like, what do you say? The word? Identified, yeah. So let's highlight that. Meow meow. Also, why not have a text box with that icon, right? Actually, I can make a sticky note. Um, so one bacterium can look very much like another. Therefore, scientists need special markers for the genes with the desired DNA, right? So with the desired DNA, wait, they need to transfer special marker genes with the desired DNA, so that they can identify the microorganisms in which the transformation has taken place. These marker genes are usually genes that make a bacterium dependent on their particular nutrient, or which causes the organism to flourish in UV light. If it's dependent on a particular nutrient, then you can grow it in an agar with nothing but that nutrient. And so if it grows with nothing but that nutrient, therefore the only one that will survive is going to be that bacterium. So therefore we know that it's that bacterium that is the one where the transformed DNA. And we can use the processing known as replica plating to identify the common cells. Okay, I'm just gonna wait. Oh my god. Oh my god. Uh, because you're making such a big deal out of it. Oh, alright.
しもし私だけど今暇潜伏えいじゃそれはやんや合わせろやねブレもしもし私だけど今どこもしもし私だけど今暇潜伏えいじゃそれはやんや合わせろやねかちょ私かよいやチャイナラブデスブブー体は猫でできているにゃにゃ死ぬかドリドリドリボコーンタイムニューイン
Some guy on Twitter says he did vocals for this track already. Should I check him out or no? Let the jury decide. Okay, I'll go look. Let's see. Come on. Dude, it's not gonna load. Maybe my bandwidth sucks. I hear the sounds of cooking. Okay, here's vocal mix. Well, dude, I'm totally gonna critique this, though, so just so you know. No, I'm not. Mother! Lindsay, come here, listen to this! Press right here. This guy did vocals for this track. Well, like, from my SoundCloud thing? Tell me what you think of this.
Okay.
And minus this, the only thing I hate about it is the stupid stuff. Check this out.
That's fast.
I hate that. Don't worry about it. I can fix that.
Sounds really good. Wow. This guy.
Okay, I gotta get a hold of this dude. Okay, he's on it. I don't care.
Sound of his voice.
Dude, kidding me?
Whoa. It sounds like the song on Tarzan.
Dude, I gotta follow him and send him a message.
Sounds like the song on Tarzan that always makes me cry.
It is so good. But he's got the theme and everything. Because it's this track, the title of it, it's off a book that or a short story that I read a while ago, and it's called The World That the Children Made. And dude nailed it. Hang on. Dude. Like, just trying to get clever there. Whatever. You have to, if you read the story, it's so good, but I expected it to be bad, dude. Well, you know what I mean? Okay, hang on, I gotta follow, dude. And then I gotta, oh, dude, better not be someone I hate. Nah, hang on, he's not, I don't know. I messaged him. If he's watching, well, he's probably watching because he just sent. Dude, can I please? Dude, let's do it. Dude, I'll give you writing. I'll give you the pub on your vocal. Dude, let's do it.
もしもし、私だけど今どこ？もしもし、私だけど今暇？潜伏？スリル？はやいや。合わせろやめか。ちょうだ。スリル？はやいや。合わせろやめか。ちょうださよ。いや、チャイナ、ラブピース。ブーブー。体は猫でできている。にゃにゃにゃ。にゃか。さあ、イエスさ、おめしゃべし、ボカーン、タイム、ニュイーン、テンペラー、テンペラー、ドリドリドリドリ、ボカーン、ドリドリドリドリ、ボカーン、タイム、ダメだというのか、ゆる、ごめん、セーブ。
That was awful. Okay. So alongside the cDNA, right, in the plasmid, we also put in transfer marker genes. This is the two things we're doing. We can also use Hasinos replicating to identify recombinant cells. Recombinant cells again, cells with recombinant DNA, cells with modified DNA, XYZ, all of them, involving growing identical patterns of bacterial colonies on agar plates with different media. This allows us to identify colonies that cannot survive without a particular nutrient. Okay, so it is involving the first case. And the second case, um, involves an identical pattern of bacterial colonies on agar plates. So you have a bunch of like identical colonies of bacteria growing on agar plates with different indicator media, and it shows the things which cannot survive without a particular nutrient. These are bacteria which have been genetically modified. These the main stages of the process are shown. So this one, method 1 as opposed to method 2, is labeled here, and how we're going to do it is going to be shown. Now, when are we going to find it? Do I know where to see it? Not really, so forget about it. Stage 1 is going to be bacteria containing all the desired gene, all contain a marker gene that requires them to require or that makes them need a specific amino acid in the nutrient medium that they're going to grow in. Let's say that nutrient or that kind of protein is going to be amino acid A. Now, bacteria and we have a bunch of identical um, colonies of bacteria growing in multiple agar plates, all of them having different ones, A, B, C, D. Now, it's going to die in all of these, but it's going to live in A. Or perhaps we could say is ones with A in it, only the desired bacteria is going to want to live in the A medium. Okay? So bacteria, including some that have been successfully engineered, so it's going to be a sample containing both the modified as well as the unmodified. The modified as well as the unmodified bacteria are going to be placed on a master plate on a complete medium. Now, the master plate is going to be inverted, pressed on a sterile velvet surface, leaving input on the colony. The master plate is saved. What is velvet? Sterile velvet squares are specially prepared sterilized cotton fabric pads used in microbiology for replica plating. Injecting the stamp to transfer bacterial colonies from a master petri dish to a new secondary plate, allowing scientists to copy extract complex patterns of microbial growth. Let me see. Oh, I see. It picks up all the bacteria. Huh. How does it do that? The synthetic fabric. What's synthetic medium? Produced by synthesis instead of being isolated from a natural source, may be identical to a product obtained. Produced by synthesis thought to have the same effect as a natural counterpart but chemically different from it. Artificial, not genuine. Pertaining to the joining of bound morphemes in a word. Okay, that's from my grammar. Synthesis is the formation of something complex before form. Yeah, reaction of hmm. Yep. Hmm. Hmm. I just want to finish this chapter. It'll be over in like it's like one page left. Yeah, yeah. If it's not over in 10 minutes, I'll get up anyway. So it's fine. So here we go. So now we have it on the sterile velvet, right? So we have an imprint of the colonies on the sterile velvet, and the master plate is saved. So now we're going to place that on another colony over here. The second plate, which is a replica plate with a specific kind of like indigo media. So this is going to be inverted placed against the velvet surface to have an imprint of the colonies. The medium of the replica plate lacks the specific amino acid required. The replica plate is going to and it's going to be incubated. The colonies that grow have not been genetically modified because they can synthesize without the amino acids which are required. So the ones which died, these are going to be genetically modified. The ones which lived are going to not be genetically modified. The ones that live. This is going to be a medium missing the essential amino acids. The replica plate is compared to the master plate. Okay, so this is going to be one with the complete medium, with the one with the amino acid, and we see that these ones grow. We're not going to be making a medium which has only the amino acid A. That's not how it works. We're going to be having one with all of them and one without specifically the one that it requires, and finding out what's missing, and those are going to be the modified bacteria. Seems kind of a sketchy way to do it, but you know, I guess you do this multiple times and you figure it out. Because you're like a gem being farmed. When you crystal and fall and goes away. Oh my god. I remember using this application before for Rocket League, dude. Oh my god, why would you do that? How do we navigate this? For the majority. Holy shit. Man, this is old school. I can see the inverted image. Okay, no, I got it. Okay. Is there any, like, what is that? Like that? Okay, that's cool. This looks nice. Man, I want to do still life so bad, bro. It's been so long. It has been a while. Oh, damn, that's pretty. Alright, We downloaded the application. We'll pay in a little bit maybe. I'll get back to this. So, god damn it. Okay, yeah. Okay, this is clear. Vectors, we gotta finish this. It's just one page. Quick. Vectors, they play a key role because we already know what vectors are because right now we're learning the plasmids. They're going to be the thing that holds the recombinant DNA that we're going to input into the host cell. It's going to be a key role in the formation of recombinant DNA as well as the genetically modified organisms. They transfer the required gene as well as any other marker genes into the new cell. I forgot about this. This is important. A successful vector targets the right cells, ensures that the desired gene is incorporated into the host genetic material so they can be activated. Desired gene is incorporated into the host genetic material so they can be activated. Well, not necessarily it's going to be the host genetic material because this was in the plasmid, which was just kind of separate, right? Does not have any adverse side effects. This is also another factor, right? Because if we deleted a part of the thing of the main nucleus, that might have been a problem. But if it was in a plasmid, which is going to be, you know, replicated and it's going to be transcripted, translated like separately as in it doesn't like affect this, then it's going to be fine. Remember that the vector cannot have any adverse side effects to the host cell, okay? It can't, or else it's not going to be a very successful vector. Plasmids are going to be particularly useful as vectors in the formation of GM bacteria and in the formation of GM plants. However, other vectors are used when we want to carry DNA into human cells. So for prokaryotes and for plant cell eukaryotes, it's fine, but for DNA into human into animal eukaryotes like humans, we want, and into some types of plant cells, we're going to have to use other techniques they're going to be having varying degrees of success. Now, personally, the only thing that I've known, the only way that I've known you do vectors um, is by bacteria, is by plasmid bacteria, right? Plasmids into the bacteria. I don't know anything else. But so this is going to be all new to me. The gene guns are used to shoot DNA carried on a very small gold or tungsten pellet ball towards the cell at high speed. Some cells survive this treatment and accept the DNA as part of the genetic material. This is so funny. It's like a cannon. Okay, how does this work? How how? Okay. Harmless viruses can be engineered. Okay, this is something that we've learned about before, where viruses which are non-lethal are going to be engineered to carry that desirable gene, and it's going to infect that animal cell, thus introducing that desirable DNA. Right? So therefore, the DNA that is going to be containing the gene that we want is going to be present in the DNA of the thing. And so when the virus then incorporates its DNA with with the host cell DNA, then we're going to be having some kind of proviral action, right? And that's going to be totally fine because the virus is harmless already, and it's going to be having that, and it's going to be transcripting and translating the gene that we want. So that could be a vector. Liposome wrapping is a technique in which the gene is inserted is wrapped around liposomes, which are spheres formed from a lipid bilayer. Okay, so we know that we have those spheres. What's it called? Spheres of them. It's called like uh, sphere of bilayer. Sphere of phospholipid mycel, right? Bilayer. Oh wait, what? Liposome versus mycel. Liposome and mycels are nanocarriers used for drug delivery, but they primarily differ in structure and capacity. Liposomes are larger bilayer vesicles with an aqueous core, while mycels are smaller monolayer aggregates that with a hydrophobic core for lipophilic compounds. Liposomes offer higher stability and controlled release, whereas micelles are better for solubilizing. Solubilizing, by the way. Solubilizing. Solubilize. Okay, buddy. Solubilizing. Okay, and offer a highly stable and controlled release, right? Okay. Lipophilic. No, that's not what I want. I want aggregate. Aggregate's the mass assemblage. What does it say? As a verb. What does it mean as a verb? Aggregates with a yes. Okay, fine, fine. I don't need to read this extra bit. Okay. Liposomes. Okay, so liposomes are basically just a bigger myocyte with an aqueous core. And so liposomes are gonna fuse with the target cell, like how vesicles fuse. This is also gonna be acting in a similar way. It's gonna be fusing with the target cell membrane, fusing with the target cell membrane. Yes, because that is also going to be nonpolar, and it's going to be allowing, and so it can inject the DNA into the cytoplasm. So it's kind of like a hormone. Microinjection, DNA injection as a way of introducing DNA by injecting it into a cell. Holy shit. Through a very fine micropipette. Okay. I was like thinking about it, but I'm okay. This is a manipulator using a micro manipulator because even the steadiest hand would tremble enough to destroy the cell. The method is not very efficient because many cells have to be injected before one accepts the DNA successfully. However, it is the method that has resulted in the most successful transgenic animals. Wow, that is so cool. Okay, dude. Mike. Yeah, man. Yeah, man. Yeah, girl. Yeah, man. Oh my god, this is a thing. That is so cool, man. That's so freaking cool. Okay, sorry, that was really fun. Okay, yeah, that's really awesome. Once a vector is inside the cell, next challenge is for the new DNA to reach the right place. This is proving very difficult, particularly when liposomes are used as the vectors. Estimates suggest that one in every 1,000 genes that enter a cell in a liposome enter the nucleus to be transcribed. Researchers are okay. So when we use liposome as the vector, it's only one in every 1,000 genes that actually enter the nucleus. Researchers are trying to modify the vectors so that new genes enter the cells and then nuclei more effectively. So far, viruses are much better at taking DNA into the nucleus than other methods because inserting DNA into the genetic material of other organisms is an important part of how viruses reproduce, right? Because like viruses are already like very adapted into doing that. However, the viruses that are used in drinks can cause an immune response in the same way that pathogenic viruses stimulate an immune response, because they are pathogens. Consequently, however, much of the research into genetic modification of human cells is focusing on non-viral vectors such as liposomes. This is because they can cause fewer side effects and potential immune responses, even though they are not effective at transferring. Okay, so that's cool. That kind of tells you about like what people are kind of researching. Knockout organisms silencing genes. Genetic modification does not always involve adding a new active gene to the genome of an organism. In some situations, scientists want to remove or silence a gene. Yes, that is true. Genetic modifying is not just adding, but also removing. So knockout organisms are widely used and are very important in genetic research. In a knockout organism, scientists silence or knock out one or more genes. They are no longer functionable. They do this by inserting a new gene similar to the gene to be investigated, but which makes the original DNA sequence impossible to read. As a result, the original gene is silenced. They cannot produce a protein. Knockout genes are usually incompleted by marker genes to show that they have been incorporated. Researchers can use knockout organisms to identify the function of the gene. I see. That's interesting. Um, Genome sequencing identifies many genes, but genome sequencing, right? Because we know what DNA sequencing is. We know DNA sequencing. We know DNA sequencing and DNA sequencing and DNA sequencing versus DNA profiling. Yes, profiling. I love profiling. Profiling the rest of the species because they are different species. Jabba-dabba-doo, jabba-dabba-dee, jabba-dabba-doo, jabba-dabba-dee, 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 jabba-dabba-doo. Okay. Scientists often do not. Okay. Scientists often do not know the function of these genes in an organism, so knocking them out and observing the results can help make the function clear. We can also use knockout organisms to investigate disease and test potential treatments. Um, Knockout organisms to investigate disease and potential treatments. We can knock out genes that are known to be non-functioning in human disease to create animal models of the disease. Wait, what do you mean by knockout genes? A knockout organism to identify is a knockout organism the same way we have like vectors, which are organisms as well? No, vectors are not organisms. I'm sorry. Knockout organisms are widely used. Um, in knockout organism science, one or more genes to know on function. This, so I'm guessing the knockout organism is going to be the GMO, right? So we're also GMOing them, GMOing them. So in the same way of using a plasmid to GMO them, we're going to be using a knockout. We're going to be using okay, this is important. So they use a new gene similar to the one to be investigated, but it has that DNA sequence, but it makes the original DNA sequence impossible to read, and so the original gene is silenced and cannot make a protein. Knockout genes are usually accompanied by marker genes to show that they've been incorporated in the same way. So this is where this is a similarity. So let's say this is the original thing, and this is the part we want to remove. And so we're going to be giving like this thing which has this part removed. It's not there. Since it's similar, okay, but it also has the bit which makes this one unusable, right? So that's what they do basically. So they kind of knock out this first one and use this one, and this one doesn't have the gene, this gene present. So it's gonna just be producing everything without that. And so when we use that, we can find out what the functions of genes are in an organism. Um, we can also use this to investigate disease and potential treatments. We can knock out genes that are known to be non-functioning in humans. Create animal models of the disease. These animal models are invaluable. Right, because we always use rats and other animals before. First we do computer testing, then we do bacteria, and then we do animal testing, and then we do human testing. And that goes in 3 phases or something. So yeah, double blind test and all that. These animal models are invaluable for progressing our understanding of human disease and ways of treating it. So true. So true. But they're so right. Okay, I'm done. Guten bye, everybody. Guten bye, guten bye, guten bye. Guten bye, guten bye, guten bye, guten bye, guten bye, everybody. Guten bye, 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 bye. Ba 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 bum 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 bum. Guten Bye, guten Bye, guten Bye, guten Bye.
Brand new.
God, scare me, bro.